mutated, either deletion or point mutation, right? Then they have zero, right? A functional gene, and that will lead to diseases like Prader-Willi, right? And Angelman syndrome. Yeah. Or if cells inherit two copies of a gene from the same parent, instead of from each parent, inherit two copies of a gene from the same parent, what we call uniparental, right? Disomy, right? And if that cell normally imprints the copies of those genes, right, from that parent, then that's another way a cell may not have, right, any functional, right, gene. Yeah, that can also be a mechanism of disorders like Prader-Willi or Angelman syndrome. Yeah. You said? Right, both copies will be imprinted, yes, and they will be turned off. Yeah. Mhm. So those are just some examples, right, of how that concept can be tested. Yeah. So a gene is chromatin, which means it's accessible. The process of what? Making a copy, right, of that gene or making a copy of the DNA, we call that replication, right? Replication. Replicate something, make a copy of. And replication is important because, right, when cells divide, they need to give a copy, right, of the gene to the daughter cells. Yeah, they must. So it's important to be able to replicate. And replication is part of which phase of mitosis? When does replication take place? Which phase of mitosis does replication take place? Which phase of the cell cycle rather does replication take place? The S phase. Good. S phase. All right. So which an S phase is between what? The G1, right? And the G2 phase. So you have the G1 phase, initial growth phase, we have the S phase, G2, M phase. So the transition from G1 to S is regulated, highly regulated. We don't, right? We need to protect the genome. We want to make sure cells only replicate, right, when the DNA is normal. It's as normal as it can be, right? So that we don't pass on mutations, right, to other cells. So that's why we have our guardian of the genome, right, mostly regulating the cell cycle. And what they do, the two major proteins we want to know at that level are what? P53 and retinoblastoma, right? Proteins. They regulate the G1 to S phase. And P53, in addition to proteins that can scan the DNA, right? It's going to scan the DNA and find out if there's any mutation. If there is, it's going to stop replication until repaired. If there's no problem, then it's going to progress. Sometimes if the repair is so bad, it cannot be repaired. If the damage is so bad, it cannot be repaired. P53 can trigger what? Apoptosis of the cell. Yeah. Retinoblastoma, on the other hand, it forms a complex with a protein called E2F. It's an eukaryotic transcription factor. Retinoblastoma forms a complex, right, with a protein called E2F. And in the phosphorylated form, retinoblastoma releases that protein. But if it's hypophosphorylated or dephosphorylated, it's going to hold on, right, tightly to that protein. But once it's phosphorylated, it's going to release the protein. And once it releases that protein, E2F will enter the nucleus of the cell and turn on the genes that will make the proteins and enzymes we need for what? DNA replication, which includes the DNA polymerase. It includes helicase enzymes. It includes single-stranded binding proteins, right? DNA polymerase 3, DNA polymerase 1, right? etc. Yeah, that are needed for RNA primase that are needed for replication. Yeah. So that's why RB and P53 are what? They are tumor suppressor genes. And mutation in those genes can lead to what? Cancers. But it's uncontrolled, unregulated, right? Yeah, cell proliferation. Yeah. Which virus makes oncoproteins that inhibit P53 and retinoblastoma? HPV. So HPV makes what? E6 and E7. E6 inhibits P53, E7 inhibits retinoblastoma. And that's how HPV is able to turn on cell cycle, right? In squamous cells, epithelial cells, unregulated cell proliferation. And that can progress towards dysplasia and eventually, right, carcinoma. Yeah. All right. So we get the S phase. Once you replicate DNA, what do we do first to replicate DNA? Now, what's the first step in replication? Replication is different from transcription, right? Replication is just making a copy of DNA. Transcription is what? Making RNA from DNA. That's what transcription is. So it's just a replication. I want to make a copy of DNA. What will I do first to transcribe DNA, to replicate DNA? Remember, DNA is a what? Double helix, right? So what is replication? You're gonna replicate, so double helix made up of two strands. Your first step is what? Separate the strands. That's the first thing. I can't do anything if I don't separate the strands. So that's why even for both transcription and replication, separation of the strand is the first step in both of them. Separate the strands so that you can use each strand as a template to make, right, another copy of DNA. Right. And how do we, which enzyme separates the strands? Helicase. Helicase separates the strands, double helix. The origin, right, and this is something testable. Typically, the origin of replication is located, right, at the part of DNA where we have a lot of adenine and thymine, right, linkages versus guanosine cytosine, right, interaction. Because how many bond is formed between guanosine and cytosine? Three bonds, three hydrogen bonds. Between A and T, two bonds are formed. So which one will be easier to break and open up? It's going to be AT, right? Bonds. Yeah. AT bonds will be easier to break and open up. Yeah. So it is more efficient for helicase to target AT rich region. Yeah. It's more efficient. So once we open up DNA, we have two strands. Each of them can be template. Each of them will serve as a template for a new strand. So what enzyme will come in next after separation of the strand? What do we do next? Okay. Even before primase comes in, you remember now you have to prevent the strands from reannealing. By open them up and you leave them there, what happens? They're going to form the hydrogen bond again, right? And close up. So that's why you have the word single-stranded, right? Binding proteins, SSBs, right? Single-stranded binding proteins. to bind to each strand and prevent them from what? Re-annealing, right? From coming back or reforming the helix. Alright. Then after that, like I said, RNA primase comes in. Now, RNA primase is a DNA dependent, right? RNA polymerase. It's gonna read the DNA template and synthesize what? An RNA strand. And the reason we need RNA primase, right, to synthesize RNA primer is because DNA polymerase cannot start from scratch, right? DNA polymerase cannot start synthesizing DNA from scratch. It needs to build on something. So that's why we're making the RNA molecule, right, as a foundation for DNA polymerase to build on. All right. After RNA prime, okay, so how does RNA primase, how does it read DNA? What direction? 5 prime to 3 prime or 3 prime to 5 prime? Hmm? So it reads DNA from 3 to 5, correct? And synthesizes what? 5 to 3, right? Reads from 3 to 5, synthesizes 5 to 3. Every time, and this applies for, just remember that every applies for replication, applies for transcription. We will always read DNA from what? From 5 to 3, and from 3 to 5 rather, and then we synthesize, right? 5 to 3. Yeah. So RNA primer will, right, read 3 to 5 and then synthesize 5 to 3 direction, a short RNA, right, strand. And then DNA polymerase will now come in, right? DNA polymerase 3 will come in, and we're describing the bacterial, right, replication. Yeah, not human. Humans, much details. So use the bacterial replication process as a model, right? Yeah, for the human one. So DNA polymerase 3 comes in, bacterial DNA polymerase 3, and it's going to build on that RNA primer and continue reading 3 to 5 prime and synthesizing, right, 5 to 3. Yeah. There are two strands being made simultaneously, right? Because we know we're starting with two template strands. So we're going to be making two strands simultaneously. One of the strands will be synthesized what? In the direction of the what? Replication fork. That is, as I'm opening up the DNA, right, molecule, that is the replication fork, right? I'm going to be synthesizing a strand in that direction, facing that, right, helicase, right, enzyme. While the other strand will be synthesized, right, in the opposite direction, like backwards. Almost, it's opposite to the replication fork. So the ones that is opposite to the replication fork, we call them what? The lagging, right, strand. The strand that is synthesized towards the replication fork, we call that the leading, right, strand. Yeah. All right. And the lagging strand is synthesized what? In short fragments. So we can notice it's not facing the replication fork. It's going to be synthesized in short fragments. Yeah. And we call those Okazaki, right, fragments. While the leading strand will be synthesized continuously, right, towards the replication fork, also called a continuous strand. Yeah, all right. So DNA polymerase 3 has 3 prime to 5 prime reading ability, 5 to 3 prime synthesis ability, and then it also has what? A 3 prime to 5 prime exonuclease. It can, on the new strand, it can go back and proofread, right? And make sure the nucleotide we are placing in the new strand matches what? The template strand correctly, matches the template strand. So if there's a mistake, it can literally look back 3 prime to 5 prime and in the 3 prime to 5 prime direction and remove the wrong nucleotide and replace it, right, with the correct one. Yeah. So it has 3' to 5' reading, 5 to 3' synthesis, and then 3' to 5' exonuclease, which is the proofreading, right? Activity. Polymerase 3. So we get to the end, right, of the DNA. We replicated the DNA polymerase 3 completed the replication. Now we have two issues we have to deal with. First issue is we still have RNA, right? Molecules in that new strand, correct? The primers. We need to get rid of them. The second issue we have is what? All those Okazaki fragments, right? We need to join them together. So in comes what? Ligase, right? To join the fragments together. And then DNA polymerase 1 comes in to do what? Remove the RNA primer through what? 5' to 3' exonuclease activity. And then replace those RNA primer molecules with DNA by reading the template 3 to 5, synthesizing, right, 5 to 3, as well as proofreading in the 3 to 5, right, prime direction. So basically, DNA polymerase 1 is the same as DNA polymerase. It has all the activity as DNA polymerase 3. It has all three activities, same as polymerase 3. It can read DNA 3 to 5, synthesize 5 to 3, and then go back and proofread, right, the new strand 3 to 5' exonuclease activity. In addition to that, it has what? A 5 to 3' exonuclease activity to remove what? The RNA primer. Is that clear? Yeah, with DNA molecule. Synthesize DNA, right? Just like polymerase 3 can synthesize DNA, right? Yeah. So it can also synthesize DNA to replace the RNA molecule. Yeah. Excellent. Another issue we deal with during DNA replication is the coiling, right? Supercoiling of DNA. So it's like you're trying to unwind telephone wire, right? As you're unwinding one end, the other end, right, will be coiling on itself, right? More and more and more. Yeah, so that same thing is a problem, right? In doing DNA replication. As helices is opening up the strands, the distal part will be coiling on itself. We call those like supercoils. Yeah, so we need to be able to unwind, right? Those the distal strands, and that's why we have what topoisomerase, right? The enzymes 1 and 2. Yeah, one can cleave. It's gonna make cleave a strand, usually one strand, and it's on twist, right? And closes this, ligates the strand back. While DNA polymerase 2 usually it can open up, nick two strands, both strands, right, to untwist the DNA and then ligate them. Yeah, but both of them require ATP. Yeah, so we have that, right, to personalize enzymes. And there are a lot of they are the target of many they are the target of the fluoroquinolones, right? Topoisomerase, yeah, DNA gyrase, bacterial topoisomerase 2, uh, uh, topoisomerase 1, also called DNA gyrase, is the target of fluoroquinolones. And also we have chemotherapeutic drugs, right, that can target topoisomerase in cancer cells. Yeah. So those are important that way. Another issue we deal with DNA replication is, now this is unique to humans. Human DNA is bigger, right? Way bigger than bacterial DNA. So if we are replicating our DNA, right, using one origin of replication, which is what bacterial chromosome, which is a circular chromosome, it only has one origin of replication because it's a small molecule, right? Even with one origin, they can still replicate their DNA in time. But humans, we have a larger DNA molecule. So we need multiple origins of replication. So DNA is replicated at multiple sites simultaneously. That is why even though our DNA molecule is large, it doesn't take too long, days. It takes hours to replicate. Yeah, because of multiple origins of replication. Yeah. Another concern with DNA replication is, with each replication of DNA, the telomere of the DNA gets shorter. With each replicative cycle, the telomere, the end of DNA, gets shorter and shorter and shorter. It's a lagging strand conundrum. Yeah. Because of that lagging strand, we lose some piece of DNA right towards the end. Yeah, with each replication. The problem with that is, imagine there is no mechanism, right, to correct that problem. So every time a cell replicates, the DNA gets shorter, chromosome end gets shorter. Eventually, P53 will see that as what? As damage, like irreparable, right? DNA damage, and trigger cell death, programmed cell death, apoptosis. So to prevent ourselves from prematurely, right, undergoing apoptosis or replicative senescence, basically getting old too soon, we have what enzymes we call what telomerase, right? Enzymes. So telomerase it carries a template. It carries an RNA template. Yeah, telomerase carries an RNA template, and it will use that template to synthesize, right, DNA. So telomerase is an RNA-dependent DNA polymerase. It's basically what? A reverse transcriptase enzyme. Yeah, it's gonna use RNA as a template and synthesize DNA. So it's gonna use its RNA template and add DNA molecule to the end of DNA, to the telomere of chromosomes, to elongate them and delay, right, that replicative senescence for as long as possible. The more replicative potential a cell has, the more telomerase the cell has. Or it should be the other way around. The more telomerase the cell has, the more replicative potential, right, the cell has. The more telomerase the cell has, the greater the ability of the cell to repair itself, or the tissue to repair itself, healed by regeneration. Yeah. The less telomerase, the less lifetime replicative ability, and and that cells with low replicative ability, if the tissue that cell makes up, if the tissue the cells make up gets damaged, that tissue will heal by fibrosis. It cannot replicate and regenerate or repair completely. It's gonna just heal by fibrosis. Once the cells are lost, they're lost. Yeah. So that, right. And that brings the concept of what? Concept of permanent cells labile cells, right? And what's the last one? Stable, yeah, cells. Where labile cells have more replicative potential. They can replicate. They are always in the cell cycle. They have a lot of telomerase. Yeah, bone marrow, hair follicle cells, stable cells, liver. Stable cells will differentiate, leave the cell cycle, but when there's damage to the tissue, they're going to re-enter the cell cycle and replicate to regenerate. That is why liver cirrhosis have what? Regenerative nodules. If you look at the histology of liver cirrhosis, there's fibrosis, but in between the fibrous tissue, what do we have? We have replicating hepatocytes in the middle. They re-enter the cell cycle, trying to regenerate and restore function. And if the damage continues and the cells keep replicating, what will happen eventually? They can acquire mutations, become dysplastic, and that will lead to what? Cancer, hepatocellular carcinoma. Yeah. Then permanent cells like muscles, neurons, once they are damaged, what happens? They can't replicate. So what happens to this location, to the tissue, to the heart? It cannot replace the muscles that have been lost. It has to heal by what? Fibrosis. Neurons, once they are damaged, what happens? Actually, the older we get, the less the ability to even to regenerate. Once they are damaged, we can't replace them, right? We the that will be replaced by gliosis or fibrosis in the brain. So what happens to the function of the brain with each loss of neurons? The function diminishes. What happens to the function of the heart with each infarction? Its function diminishes, right? Right. So in a condition called progeria, where patient presents with what premature aging, right? A 13-year-old that will look like a 90-year-old man. One of the mutations that can lead to that is damage to the mutation affecting telomerase enzyme. Yeah, so they have no telomerase, which means the ability of the cells to replicate is limited, and the patient cells stop replicating. They grow old, right? Fast or earlier than normal. Yeah, all right. That's why there's no cure for aging, right? Aging can never be cured. All of us will grow old and die. Botox to take care of the outward appearance, right? Yeah, I mean, I'm 70, but I can look 50, right? Yeah, for as long as possible. But eventually, what about the internal organs? Uh, that's right. Right. Yeah. And the failure of the internal organs is actually what kills, right, during natural aging, not failure of the outward, right? It's the vital ones that we cannot slow down. Yeah. All right. So that concept is there. Then cancer cells are like super lipid cells, right? Cancer cells have a lot of telomerase enzymes. Yeah, they can replicate basically for life. They don't stop. They can keep going, keep going. So that's why they are so aggressive. And that's why we target cell cycle, right? Drugs that target cell cycle to just shut down, right? Yeah, their replicative ability and slow down, right? The spread of the tumor. So yeah. All right. So that's replication, right? What is PCR? Polymerase chain reaction. What is that? PCR. I'm sure how many of us have done COVID PCR test? So what is PCR? Right. So you're looking for the genome, right, of COVID virus. Because if we the key is there. So a patient has COVID infection, and viruses are small particles. They're gonna make some DNA, etc. But we need to be able to. We may not be able to see it very well. Like we won't be able to detect them. They may be small, right? To be insensitive, right, to the test or machine we're using. So you use PCR to blow up, right? It's like a way of amplifying the genome. You want to blow up the genome, make it like millions, make millions of genomes. So it's going to be easier to detect, right? Those genome, right, in the sample that we have from the patient. And that's what PCR is. So PCR is basically making copies of genome, DNA. Sometimes you can also do that with RNA. If you start with RNA, you can convert RNA first to DNA and then blow it up. But if you're starting with DNA in the initially, we just call that PCR. But if you're starting with RNA, convert it first to DNA, we call it reverse transcriptase, right? PCR. Reverse transcription PCR because you convert DNA, RNA first back to DNA and then you blow up the DNA. So PCR is the replication we just described now, right? Is what we're doing in PCR. Only you're doing this in the what? In the lab. In vitro, right? Not in vivo. Yeah. What we just described, the steps are the same. So we take the DNA molecule, what do? It's a double-stranded helix. You separate the strands. Also in the lab. How do you separate the strands in the lab? We don't have helix. Eat it up. Good. So you eat it up. You break all those bonds. Yeah. The more triple bonds the DNA has, the higher temperature we need, right? To separate the strands. But if it's made up of a lot of AT region, it's going to require lower temperature to break it up. Good. So we eat it up. Now, what do you do to prevent it from reannealing? So we heat it up, we break the strand. What can you do to prevent it from reannealing? Keep the temperature at. Yeah, because if you cool it, it's going to reanneal. Do you get what I mean? Right. So you have to keep it at that temperature to make sure it doesn't reanneal. So what do I do next? I've separated these strands. What do I do next? What do I add to the test tube? Right. So then what do you do next? Yes, yes. We separated the DNA molecule. You have your template strands in there in the test tube. What do I do next? Which enzyme do you add? Right. So we don't have RNA primase. We cannot add RNA primase, but you can add what? RNA primer. Yeah. So you can now make, instead of adding RNA primase, you make the primer yourself in the lab. You can make the primer, RNA primer. You're gonna make it because once you know the DNA sequence that surrounds the gene of interest. If I know the DNA sequence that are around the gene of interest, I can make an RNA primer molecule in the lab to complement the DNA sequence, uh, and then I'm going to add that to the test. You add the primer to the good. So you are the primase. You are the one making the primer. You just add the primer to the test tube. Now, once you add the primer to the test tube, the primers will go and bind right to the adjacent molecules. Right now, we're ready. So what do you add next? You had no. What would be the proofreading step? What would do that? You can do that. You had the primer. What would be on the primer? DNA polymerase. So you're adding DNA polymerase next. That's what you're adding. So you can, right, you had DNA polymerase enzyme next. You had heat sensitive. No, heat resistant, not sensitive, resistant. Because remember, we heat up the test tube, right, to separate the strands. So you cannot, remember, enzymes are sensitive to heat, right? They can get denatured. So in that case, you're adding a heat resistant DNA polymerase enzyme to the test tube. And what would that? And then in addition to that, what do you add? In addition to that, you've added DNA polymerase. What will you add? How will it make DNA? It needs what? Building blocks. So you now had what? Nucleotides. ATP, GTP, CTP, TTP. You're going to add them to building blocks. Then DNA polymerase now can now use those building blocks and build on what? RNA primer and synthesize the new strands. Yes, in there. Now, once you do that, what do you do next now? You now cool the sample so that what? The new strands and the temperature can now form what? Reform the hydrogen bond, right? Yeah. So we started with one DNA helix. After the first round, of after the first round, you heated, separated RNA primer, DNA polymerase with all the nucleotides. We synthesize and we cool. At the end of that, when you cool, when you check the sample, you're going to have what? Two, right? DNA molecule. Yes. You start in the next cycle again. Repeat the same thing we just did. Heat up the two strands, separate them into four single strands, add four RNA primers, right? And then had four DNA polymerase enzymes, and then had as many as much nucleotides as possible, and then you cool again. And then what do you have now? You're gonna have what? You're gonna have two times two with double, right? Four strands. Yes. And then you start another, that's what we call PCR, polymerase chain reaction. So you're going to be doing that thing over and over and over again. So you can keep growing up DNA, right? Keep doubling until you have millions of copies of DNA. And we can now take those samples now, put them on a what? On a electrophoresis and separate the strands and then stain them, right? So you can identify what we're looking for. And that will now, if I now see COVID genome, I will now know the right patient has COVID virus infection. If I cannot see the COVID genome, then I know patient is negative. So the PCR is basically the in vivo DNA replication process I just described, right? But it's taking place right in the lab. Yes.